effort. So I would like to start by introducing Dr. Emily Miller. Dr. Miller, her talk, I heard about the research she does, and I thought that would be perfect for changes. The topic that we are exploring through this whole speaker series is about changes in Monterey County habitats over time. And the question comes up, how do we know that there's changes in these habitats? And so Dr. Miller's research speaks specifically to how do we know there's change and how do we document that change? She is currently with the Monterey Bay Aquarium Research Institute and has also worked with the Aquarium Ocean Memory Lab. And that alone is kind of an intriguing title there. So Dr. Miller, I'll just let you explain to us, how do we know there's been change in the marine habitat?
Yeah, that's a great topic, and hopefully I'll cover a bit of that now. Let's see. All right. Thumbs up if you can see my screen. All right, thank you. Okay, so I'm going to talk about marine algae as ocean record keepers. So one way we can know about change is by actually looking at the seaweeds around us and in the Monterey Bay. So the idea behind this study that we did at the Monterey Bay Aquarium is that we need observations to make predictions about our future environment. So this example here on the right, Arctic sea ice is dwindling. In the figure, the blue and the red lines with the confidence intervals around them on the right-hand side are two different scenarios. So estimates about what could happen in our future with sea ice. The purple, which is just that little section in the middle, is our observed data that we have. And the brown is also modeled. So like the future, the brown on the left, the past is also modeled, because all we have is that little purple section of our timeline where the data is actually recorded, where we have observations. And so if we could extend this recorded observations further back in time, we could make these more accurate predictions of these future scenarios. And the reason why we only have this very small section of recorded data is that instrumentation only began in the latter half of the 20th century. So we're only able to record our environment in this very short window of time. So it's not too much data to be able to document change and then to make these predictions about our future or even predictions about our past. But we don't just need instruments to be able to record information about our environment. Living tissues themselves store information about the environment. So this is a cypress tree in Virginia, and this cross section of the tree rings can indicate the precipitation that occurred each year of a tree ring. And so there's these actual historical events in the colonial era of that region that are noted here on the tree rings that we know happened, and then we can see them happening in the tree rings themselves. Another example, using stable isotopes, we can actually reconstruct communities of organisms, not just now, but in the past. These paleo communities, each sort of shaded area is a category of animal in a food web. So there's herbivores in green, there's faunivores or carnivores in blue, and there's omnivores in tan. So we're able to take tissue samples from preserved animals and reconstruct sort of how these animals interacted in a food web in the past. So the Monterey Bay Aquarium's Ocean Memory Lab, that's sort of the idea behind it, that we can unlock the data about environment from within animal, plant, algae tissues themselves, that they are actually recording the memory of the ocean, since we're focused on the ocean here, in their tissue throughout time. And so some examples that we have in the lab, on the upper left is a bulla, a whale inner ear bone. It's like this big. It's pretty large inner ear bone. That's a gray whale there. And so that one whales live a really long time. Different species have different lifespans, but these whale inner ear bones are recording, just like the tree rings, they're putting down layers each year on that bone. And so we can go back and sample over time those different rings to understand how the environment changed within that animal's lifespan. And for whales that have these long lifespans, some whales, this could be a 200-year data set just from one single individual. And sort of another approach is not just looking at one individual over time, but looking at many individuals that were sampled across time. So in the lower left, there's seabird feathers. And one study that was done at the Ocean Memory Lab in partnership with the Bishop Museum in Hawaii took all these, there's a set of all these taxidermied birds that were in the museum and had been sitting in shelves. And they took a feather from each of different birds that were in a time series, and they were able to see how the food web changed in the Pacific Ocean over 150 years. And so that's the idea that we are approaching with seaweed on the right of all these pressed seaweed specimens, where we're stitching together a historical timeline using different individuals that were collected throughout that time. And so where this sort of began is the Stanford's Hopkins Marine Station has a collection, a herbarium of pressed algae, macroalgae, called the Gilbert M. Smith collection. And this goes back to 1892 when the Marine Station was founded in Pacific Grove. And this is right next to the Monterey Bay Aquarium. And so that was sort of the start of this idea of, oh, we actually do have this historical data set all the way back to 1892 that we could look at. And there are herbaria across the world. So Stanford's collection is unique for our area, but this practice of collecting seaweed and pressing it became very popular in the Victorian era in Northern Europe and then North America. And so that's where there are many collections. And it's kind of a unique area of natural history. Women were shut out of academia in that time, but natural history involving animals in that period was generally just hunting. So they're collecting specimens by hunting, and so that wasn't seen as very ladylike. But collecting and pressing seaweed was something that was socially acceptable for women to do. And there's some beauty to the pressing as well. So it was sort of a hobby of many people, many women, especially like middle class in England that had some leisure time. And so Margaret Gatty is one example. She published this herbarium, this collection of British seaweeds, and it was the authoritative text of the time. And she still encouraged women to collect seaweed, press seaweed, but sort of within the confines of their socially accepted gender norms. So she was encouraging, still wearing your skirts and your corsets, but maybe taking a gentleman out there to under the protection to keep an eye out while you're collecting seaweed. So it was an acceptable form of natural history for women. And these are some images from her collection that are really both beautiful and incredibly valuable in the scientific approach to examining seaweeds as well. And so these are a few images from the Pacific Grove Natural History Museum's collection. So many of these seaweeds that women were collecting were then bound in these beautiful books. And this one's called Flowers of the Sea here. And they're really lovely and actually like scrapbooks. So there's some information of this beautiful microcladia that is spread out. It's really well done and a well-pressed specimen, and some information about where it was collected and what we know about this collection. And then this example is actually bound in whale vertebra, which is pretty impressive in itself. And so there are just a lot of beautiful examples that were collected by women around the turn of the last century. So the herbaria that we used in our studies, the Monterey Bay Aquarium was founded in 1984, and seaweed was collected and pressed kind of sporadically since then for a couple different projects. And so we had some that were sort of the more recent era from the aquarium, and then the Hopkins Marine Station filled out the bulk of our collection from 1892 to 1980, when they sort of, the professor that was sort of leading that retired. And then we filled in some other gaps because some of the timeline had some gaps. And so UC Berkeley, San Diego Natural History Museum, University of North Carolina, and University of Michigan all had some specimens that we were specifically looking for, a specific species, and certain years that we needed that were able to fill out this collection. So we were able to do this because there's an online repository called macroalgae.org, where you can search and figure out if you're looking for a specific species or location. You can find all the specimens that may be spread out throughout the country, really, depending on how different researchers traveled and returned to different universities. And so in the Hopkins collection, there are some really, really gorgeous examples. These are all by Mrs. Swanton. And so these, the women, these are all, they don't have exact dates on them because a lot of the women weren't collecting in the same way that academics were collecting. They weren't writing always the exact year or location. These all say Pescadero, California, which is just north of Santa Cruz. But we don't have the exact year, and it just says Mrs. Swanton. So Fred Swanton was the mayor of Santa Cruz. There's Swanton Road. Fred Swanton's known for the Santa Cruz Beach Boardwalk and getting that really up and running. So it was likely his mother, who did live in Pescadero for a time, or his wife. And the timeline could be either of those two. But a lot of the women are lost from history because a lot of our collections are based on early academics. And one that was really easy for me to track down was this Bradley Davis. I saw that in 1892, we had some species that were, some specimens that were collected by Bradley Davis. The one on the left is 1892. And then again, I came across more of them in 1922 that had his same name and a lot of the same species. And so I was really interested in that because I thought, 30 years apart, who is doing this? And there wasn't anything in between. And so I was able to find his name quite easily, whereas I couldn't figure out which Mrs. Swanton was the one who collected those others, because he was a student at Stanford in 1892, the inaugural class. And then he eventually became a professor at Michigan. And so when he was a young professor at Michigan, or youngish, he must have traveled back and collected these specimens again and left them for the Hopkins Marine Station to have. So the value of collecting is really impressive to see, around 100 years ago. And he went on to be the president of the American Society of Naturalists. So he is really well documented. And that's easier to find some of those academics and information about them. But one woman who is very much present in this collection and was not lost to time is Professor Isabella Abbott, who was a professor at Hopkins Marine Station. She's since passed away in the last several years, but she was a professor especially the 60s, 70s, before she moved back to her native Hawaii. And she is responsible for a lot of the collection that we ended up using. And so without her, this would not be possible. And she's the one who literally wrote the book that is used for seaweed identification along the Pacific coast of California. And so the objectives for our study specifically were to demonstrate a new value for unused but widespread herbaria. So these have been collected for a really long time across many institutions in much of Europe and North America. but it's not necessarily a use that has been extensively explored. So we wanted to show this new value for these collections. And we also want to generate ecosystem records before instrument measurements. So like this idea we were talking about in the beginning, we only have data about our environment going back to the mid-20th century for a lot of our instrument records of interest. And we also wanted to establish repeatable methods for wide application. So we wanted to sort of present this as a case study so that other institutions could do this and sort of adapt it to their own questions that they're interested in studying. And finally, we wanted to encourage institutions to change herbaria operating procedures to monitor global change. Now, this is not something that is new, really, to a lot of curators, but a lot of times institutions are not funding these collections so that we can continue collecting. Like, we often have these collections sitting on shelves, and in order to monitor global change in the future, we really need to continue collecting. So we wanted to make a plug for maintaining and continuing collections. So our study had 2 phases. The first phase was an experiment. So we have these historic specimens. This is a Macrocystis, giant kelp from 1892. And can we actually just sample this tissue from over 100 years ago? And does it represent actually what the ocean was like at that time, or has this specimen degraded over time? So we needed to first test, is there an effect of storage on the seaweed microchemistry, so that we could then go back into the historical specimens and be able to interpret them correctly. So the phase 2 was this historical case study. So we wanted to use these historical herbaria to reconstruct environmental features prior to instrument records. And we could only do that if we knew that the effect of storage and pressing and the paper itself didn't have an effect on the microchemistry. So for our first phase, we went out and we collected fresh specimens right off Hopkins Marine Station in Pacific Grove, so southern Monterey Bay. We were interested in covering the diversity of macroalgae. So we got 2 species that were green algae, 2 that were brown, and 2 that were red. So those are the 3 kind of main taxonomic groups within algae, even though the phylogeny of macroalgae is quite complex, but we tend to think of them as green, brown, and red. And then within those 3 categories, we wanted to get a branching and a bladed example in case the morphology affected any of the microchemistry. So we selected 6 species. We also made sure that these were well represented within the historical database. So this is just a snapshot of the database, so that we knew we had examples of these that went back throughout the time span of the collection. And so just to give you a picture of what they looked like, this is Cryptopleura, which is this beautiful bladed red algae. Gelidium, which is this kind of lacy branching red algae. Cladophora, which is like moss. So it's kind of this tight little branching green algae. Ulva, which is also known as sea lettuce. And so it's these broad blades. And people do eat sea lettuce fresh in salads. Sylvicia, which is a brown algae, and it's this branching rockweed. So it's one that's kind of at a higher tide line. So you'll see these rockweeds along the rocks quite often, even when it's not too low of tide. They can tolerate being outside water for a while. And then Macrocystis pyrifera, which is giant kelp, and it's the symbol of the Monterey Bay Aquarium, so we couldn't not do it. And it's also the big defining species of our ecosystem that supports all the otters that we love to look at in the Monterey Bay. So we collected these species. We rinsed them, and then on the left, that's Sue Lisson, our co-author, who is pressing them. And so the pressing process, we pressed them on modern paper. That's this, archival, acid-free, museum-grade paper. And then we also pressed each species on paper that was from the 1930s. And we wanted to see if this historical paper had anything in it that affected the microchemistry. So that could be possibly heavy metals from manufacturing, or the fact that they had some acids that could possibly affect the algae that's pressed on there. And we weren't really sure what we would find. So we wanted to compare sort of modern paper and historical paper. And then we also wanted to look at the effect of this long-term pressing and drying. So this is the traditional way that we press seaweed, but we wanted to see if that differed from when it's freshly pulled from the ocean and just oven dried. So on the right is an image of the algae that's being dried in the oven. And so we dried just a little bit of each specimen in the oven, and then we sampled that immediately the next day. So we could see sort of the fresh signature right away. And then we compared it to the pressed specimens. And so we processed these in the Ocean Memory Lab. And so we had them pressed, and then we were sequentially removing chunks from the same specimen over time. And then we were grinding it with a mortar and pestle down into a powdered form, homogenizing it. And then we sent these off to labs, 3 different labs for different analyses of microchemistry properties. So amino acids, heavy metals, and stable isotopes. So those were our 3 common categories of analyses that people might be interested in. And so we wanted to see if there was any effect on any of these 3 metrics. And that's, several amino acids, like 16 or so amino acids, looking at 8 or so metals, and 3 different stable isotopes. So we're trying to look at all of the different ways that the microchemistry could be affected by this pressing process. And you don't need to pay attention to the details, but each little square, each panel is a plot of an amino acid over time, and each point is a different seaweed specimen. And the basic take home from this is these are mostly flat lines. So on the x-axis of each of these is 365 days. We're repeatedly sampling them over time, and there's not much change over time. So the amino acids were not affected by the drying process. The 16 panels at the top of A are the older paper type, the historical paper, and the 16 panels B down below are on modern paper. So there's also, if you look at each plot, so like alanine and arginine, those are the top 2 amino acids there. You look below, there's not much difference in A or in B. So we didn't see an effect of the paper type either. So no effect of the paper on the amino acids, and no effect of the drying time over 1 year. Heavy metals, we had a similar result, but there was more variability, especially for certain species. So we have these, I guess 9 heavy metals. And there was some variation within a specimen over time. Like Macrocystis, the giant kelp, was pretty variable, especially in, I think, well, in several metals, but that means that it's just storing its the metals in different parts of its tissue. So there's some interesting species-specific patterns that we saw, but overall there was no effect of the paper, and there wasn't that much change over time. There was variability within our time scale, but the starting and ending points were not different. And stable isotopes over time, the top is carbon, the middle is nitrogen, and the bottom is oxygen. And carbon and nitrogen really didn't differ over time. So this is the ratio of a heavier isotope to the lighter isotope form. Oxygen, we did see sort of this slight increase over time, and that's likely because moisture, water in the air, is actually changing the isotopic composition of those specimens. But again, no difference in paper type, and over time, no change for carbon or nitrogen and very little for oxygen. So our overall results for the first phase, our experiment, did the historical paper alter the microchemistry of the pressed macroalgae? No. So we can interpret historical specimens without calibration. Did the pressed seaweed values change over 1 year? And no, but we did find some species more variable for certain metrics. So the results from this experimental phase helped inform the selection that we made for the second phase, where we were using just one species as an environmental proxy, or a ocean memory recorder. So in our second phase, we chose Gelidium, which was the branching red species, and that one was very consistent over time. And we chose nitrogen stable isotope ratios. We thought it would be one that might be interesting to look at in the system. And the isotopes analyses required like 5 to 10 grams, 5 to 10 micrograms. So a very, very small amount of the specimen. And so we didn't want to consume too much of these really priceless historical specimens. And so it was a good analysis for us to choose. Heavy metals required quite a bit more, whereas these were just taking a very tiny tip of a specimen and really not noticeably altering it at all. So then we dove back into these historical collections to get all of the Gelidium that we could from this timeline. And then we looked at several different environmental parameters as correlates. And the one that really stood out was upwelling. So the way that we measure upwelling is traditionally Bakun's Upwelling Index, which started in 1946 and is calculated using wind speed. And so we plotted these stable isotope nitrogen values that we got from the Gelidium against the upwelling index for that year that the Gelidium was collected for each species. And we, found a strong correlation. So what is the upwelling index? It's measuring upwelling, which is the process by which this deeper, colder, nutrient-rich water is pushed from offshore closer up to onshore, from the deep and to the surface. And that nutrient-rich water then just really is what allows the bottom of the food chain to grow and flourish, and then support all the other higher levels of the food chain. So, sorry, you might be hearing my 4-month-old right now who's trying to go down for his bedtime. And so that is really the reason why the Monterey Bay is as productive as it is now. It's the determining factor of our productivity. So it's not surprising that the upwelling signature would show up in seaweed. So the right half of this figure is upwelling that was recorded from 1946 on. And then we took that relationship that we found between upwelling and the nitrogen isotope signals to then hindcast, estimate for the past what the upwelling index would be based on the seaweed specimens. So on the left half of the figure, every single point is one seaweed specimen that we collected and what the upwelling index would be based on its nitrogen isotopes. And so we found this pattern that had this kind of dip in the middle of the century. And so we were interested in this dip and what was going on in the system during this time, starting in the late 1930s and into the late 1940s. So it helps to know that in the Monterey Bay, we know that there was a huge sardine fishery, and there's an anchovy fishery right now as well. And it was researchers at MBARI led by Francisco Chavez that really honed in and found out that there was these regime shifts between these forage fish species. So these are forage fish, both sardine and anchovy. They're kind of the bottom of the food chain, and they really support the larger megafauna that we have in the Monterey Bay. And what they found was that it wasn't completely random that we would have, some good sardine years and some bad sardine years, some good anchovy years, some bad anchovy years. It was actually these entire regimes where each species was more dominant in the system that would last several decades, and that these would alternate. And it was based on environmental features of the entire ocean basin, the entire Pacific basin. And so that's happening during this period that we were interested in. And we found that, so the historical sardine boom for Monterey then was followed by a crash that was written about by Steinbeck and is pretty well documented as far as shaping Monterey's history. And this decrease in upwelling correlates with the peak in sardine catch and then is followed by the shift to an anchovy regime. And so we were curious about this, but the mechanism behind it is that upwelling, when it's weak, it actually produces small prey, so small little invertebrates and little fish that are what sardines specialize on. And strong upwelling favors large prey for anchovies, more near shore, as sardines are shown here more offshore. So this weak upwelling that we found during the same time period as the sardine crash makes sense. And the entire theory by Chavez et al. Between these shifting between sardine and anchovy regimes is that in a sardine regime, the water is warmer and we have this weaker upwelling in our region. And so what we found, what the seaweed showed, fits into this general understanding of these basin-wide patterns. So in summary, macroalgae, seaweeds, revealed historical upwelling patterns before we had instrument records that were able to document it. And these environmental processes may have contributed to the sardine anchovy regime shifts. So it was originally thought that overfishing caused the sardine to crash until researchers discovered that it's actually, sure, it was overfishing as a contributing factor, but there's also these ocean basin-wide temperature oscillations, these patterns that shape the shifts between these regimes of sardine and anchovy. And what the seaweeds show is that there's also these more local processes, upwelling, that may be kind of pushing the direction of these shifts as well. So it might be all three of these contributing factors that resulted in the collapse of the sardine fishery and the shift to the anchovy fishery. So in conclusion, this method can be used by other researchers to answer environmental questions using herbaria throughout the world. This is these are collections that are widely available and go back in time, sometimes to not just to the mid 1800s like ours, mid to late 1800s, but even further. There's some in Europe that go back to the 1700s. So there's a lot of interesting resources that haven't really been mined for some of these questions. And herbaria are underfunded and underutilized in conservation research. So they are often used to look at the change of different species over time, looking at their genetics. The distribution, where they were collected over time, can show shifts in species over time. But there's still more questions that we can ask that's actually recorded in the tissues themselves. And continued collecting is necessary to provide long-term records for researchers of the future to ask questions that we can't even imagine today. So Mrs. Swanton and Bradley Davis, I'm sure, had no idea that any of the species that they collected, specimens they collected, would be used to answer these sorts of questions. Isabella Abbott may have had a better idea. But right now we can't even picture what could be asked of these collections in the future. And so there's value in continuing to collect and to document global change that's happening now. And so just wanted to thank Monterey Bay Aquarium, who funded this project. There were a lot of people that helped collect and prepare specimens in the lab. And then it wouldn't be possible to do this study without the other herbaria that contributed specimens that filled our gaps. So just wanted to thank all of them, and thank you for having me. And I can take any questions that anyone has now.
Thank you. That was super interesting. And like I predicted, right in line with this idea of change over time in these habitats. That was really cool.
Great. Yeah. And this is a specimen of Macrocystis, of giant kelp, that was used to inspire the Monterey Bay Aquarium's logo. this exact one. So
is that part of the aquarium's collection in the herbarium at the aquarium?
Yeah, I believe so. I haven't actually seen this one in person, but yeah, I think so.
Super. Other questions?
Yeah, go for it.
Yeah, go for it.
Emily, this is Tom Murphy. I was interested in your use of the Bakun Upwelling Index. It's, of course, widely used and well thought of, but it does have some shortcomings. You mentioned one of them, that it only goes back to 1946. There are some alternative data sets that you might want to consider using that go back to the late 1800s that are probably in many respects better than what the winds that the Bakun Index is based on, even from 1946 onward. One that I'm thinking of is called the 20th century reanalysis. And it would provide not the upwelling, but the winds that are used to calculate the upwelling. So you might want to think about, look into that as an alternative to extend back that index and see how well you get relationships between your biological variables and those physical variables.
Yeah, I think there's a lot more that we could do with this, and I think we will. I think this is really just the beginning. So we went for a simple index that we could easily pull.
Sure.
But we're also interested in looking at possibly ocean acidification, temperature changes. And so, yeah, I think that there are better data sets as you describe, and just sort of more interesting environmental questions that we can ask. I think this is just a first step. And yeah, there's a lot more out there.
Right. Thank you.
That was my question, is what are some of those questions that you think of that we could use Herbaria to answer? Yeah.
So the isotopes, stable isotope analysis has been used to look at ocean acidification, global ocean temperature increases. And I think there's other options, sort of, I think heavy metals could also be interesting to look at, sort of change in human land use along coasts over time, as well as amino acids are often used to understand wastewater inputs into systems, and sort of their influence on seaweed. So we could answer a lot of questions about human settlement on coastal areas using seaweed.
Chris Essert has a question about the methods that were used, and the question has to do with mass spectrometers, where they used to measure those stable isotopes and stable isotope ratios.
Yeah, yes. So for stable isotopes, we sent them to UC Davis Stable Isotope Facility, and they did mass spec on them. Yeah.
Super. Can you stop sharing so we can see everybody's faces? That helps to be able to see their hands. Feel free to raise a hand and ask a question. Type one into the chat, and this is a small enough group, we can get an interesting conversation going with any questions people have. I think the question of temperature and fluctuations in temperature in the bay is a really interesting one, particularly when we look at some of the kind of strange phenomena that we've seen in recent years with the big blob of warm water and those kinds of things offshore. And it's interesting to think of how we might use algae to kind of look at that historically. Is anyone doing that work?
Not right now that I know of. I think part of the issue is that our data set was fairly coarse. So we had specimens that were collected, some we had a few per year, but then there's several chunks of time where we didn't have any for a couple years. So we could ask these kind of questions that looked at really dramatic influences like upwelling. But something that would look at a change that happens within just 1 year you would need sampling repeatedly throughout that year. So the collection itself does limit the types of questions that we can ask and the fine scale nature of them versus sort of these broader questions.
Mm-hmm. There's a question from Tama that asks about the anchovies and sardines, and I also was not aware that the smaller fish eats the larger larger algae, the larger fish eats the smaller algae, and how that cycle happens. That's interesting.
Yeah, I mean, I don't know exactly every reason why. I think eye size is part of it, relative eye size for each species. And I also am painting like a broader brushstroke than is the full picture. They are adaptable and will move to find different prey, different size prey in different locations. But those are the sort of the general, broad brushstroke differences.
Excellent. Yeah, go for it.
Emily, there's another Emily whose work I really like, as well as liking yours, and you may know her, Emily Osborne. She's done related work. Do you know her work on using planktonic forams to infer upwelling and acidification in coastal?
I know her name and I've seen some of her work. Yeah.
Yeah, it's really when she came out with a paper a few years ago on that topic, and she was looking in the Santa Barbara Basin. I thought, wow, this is a really nice link between chemistry, biology, and physics. And your work reminds me of hers. You're using different species, but you're in many ways pursuing the same kind of problem. In her case, I forget how far back she was looking, but maybe 100 years or so, similar to what you're doing.
Yeah, it's interesting to see different ways people are getting at these same questions, but using sort of different collections and different ways of getting at it.
Right.
Carla has a question.
I do. I want to thank Emily for sharing her evening with us. And I just learned about 100 things that excite me. So thank you so much. In terms of museum collection preservation moving forward, are there any techniques or things that you would have some advice about? Like, don't preserve algae like that, or, so that you can derive these things and have better samples, assuming that the people handling the collection had a choice.
Yeah, that's a great question. Basically, our experimental part of the study found that the specimens are fairly robust, so that's great. That the paper is not really affecting any of the microchemistry. That the time frame, at least for the time frame that we looked at, did not really alter it. And so I'd say, I think really only if you're interested in the stable isotope of oxygen, that's one where it would make sense to keep everything in like a climate controlled facility that's monitoring the levels of humidity. So that was the one thing that really had an effect on the microchemistry over time. Yeah.
Chris Essert notes, as a scientist would, that of course collecting all that metadata about where was it collected, when was it collected, who collected it, how they collected, that going with the specimen is super important. I imagine you ran into difficulties with that, didn't you, Emily?
Yes, so we couldn't use all the specimens that were pressed. That's an excellent point. Thank you, Chris. And we could only use specimens that had the date. We were also trying to make sure that a seasonal effect didn't influence the collection. So we needed an actual date, not just the year. And then we needed the location. We only used specimens that were in Pacific Grove to Monterey. So from Point Pinos, if people are familiar on the Monterey Peninsula, to downtown Monterey was our exact study area. So we needed people to be fairly specific in their records so that we could use them. And luckily, many were. So that helped in doing the online database search to track down some of these specimens, and why we found that Bradley M. Davis had taken some of his specimens back to University of Michigan when he was a professor there, and had kept excellent records. So yes, time, date, and location are critical.
Yes, go for it. He's really into this.
So have you talked with the people who are in charge of collections at natural history museums around the country? And if so, are they interested in learning from what you've done? And sort of following up on Carla's question, are they thinking, how can we use the collections that we have? How can we do a better job of collecting in the future?
Yeah, I mean, every curator we reached out to for using specimens in this study was incredibly enthusiastic and wanted to be a part of it. So while we were destroying a small portion of each specimen, because it was small and didn't affect sort of the overall look of any of the pressed specimens, they were very much on board and absolutely wanted to answer more questions with the collection that they have. I think the obstacle is definitely not curators. Curators value their collections and want to use them in any ways that they can. It's more so any institutions that are not funding those collections, because they really, you hear these, to me, horror stories of these collections that are at some universities. If they aren't being funded, they can be really tossed out. There are some horror stories of that. So the value of these collections needs to be emphasized to larger institutions to which they belong.
Do you know of anyone doing similar work on plants, terrestrial plants and ferns and flowers? I mean, we have all kinds of things in our collection.
Yeah, not exactly what we are doing, but UC Santa Cruz is doing a lot of kind of historical and global change focused research on their specimens at their natural history collection there.
It's interesting. And I just have to know, what can a whale ear bone tell you? Those are the coolest thing.
Yeah. Well, a lot of the same things that we're looking at here. So heavy metals could be examined in there, and stable isotopes that look at some global ocean temperature and climate change.
Super. Any final questions? We've gotten our questions in. Does anyone else have questions for Dr. Miller and would like to get those in before we close? You have one more. Okay, we'll let him have one more.
All right. This is a little bit of a story. When I was a teenager, I went to school in San Francisco, and we got to take a trip to the Cal Academy of Sciences herbarium. It was the first time I'd ever been to an herbarium. I thought it was wonderful, amazing, although it didn't smell very nice. It smelled like mothballs, but it was fascinating to see what had been collected from all over the world. And then I remembered when I was a kid in Hawaii, we had a science project where we collected seaweed and did our own pressing. Anyway, I mentioned all that because I'm wondering, does Cal Academy have anything that potentially would be useful? I would have thought they would have some seaweed collections.
Yeah, they actually, so they do. They have extensive collections, but a lot of their seaweed collections have been sent over to UC Berkeley And that there's sort of a trade of taxa, perhaps, like, and UC Berkeley has been sort of the main Bay Area seaweed herbarium. Yeah.
Kind of a specializing.
Yeah, I think so. Yeah, seaweed often don't get as much attention as they deserve, I would argue.
Flowers and things.
Yeah.
Well, thank you for giving it the attention it needs and for giving us a new perspective on how to look at change over time in our ecosystem. Super cool talk. I appreciate it. And the museum is now open and exciting change is happening on a daily basis here. Julia is going to fill you in on what's going on in the museum.
Cool. Well, just really quick, I did want to say, Emily, thank you so much for being here. Yes, thank you for joining in our talk. That was incredible. And I hope everyone enjoyed it. I heard you mention you have a 4-month-old and I have an upcoming 5-month-old. So, hey, mom life. But thank you everyone for joining as well. Just a few highlight events that we're super excited about is this Saturday is Science Saturday, and we are celebrating Day of the Dinosaur. So to celebrate, we're doing this hybrid virtual / in the museum with reservations event. So at 10 a.m., we're doing a live tutorial of drawing a triceratops. And I know, really excited. And so we brought in the paint paddock, and he will be demoing this tutorial at 10 a.m. live. And then on May 27th is our next Hardcore Natural History event. So registration for that is now open. We will be chatting with Natalie Olivas, who is the community organizer at Regeneración Pájaro Valley Climate Action. So if you're interested in either one of those or both, which I hope you are, I will go ahead and enter those into the chat now. So you'll have links to that there. Also, just note this has been recorded. So if you're interested in the recording, let me know. We're going to work on some edits and we'll most likely go ahead and put it on our webpage as well. So I'll send a note of that once that's ready. Other than that, we are open now. So check out our website, reserve your tickets Friday through Monday, 10 a.m. to 4 p.m. as well. And I hear the aquarium is reopening as well. So super exciting there. It sounds like early May, they're doing member nights or member events from May 1st till May 14th, I believe. And then right after that, you're opening to the general public. So feel free and check that out as well. And thank you everyone for being here. Really, really appreciate it. Carla and Tamla, so glad you could also be on the call as well. Thanks so much, everyone. Have a great rest of your evening. Happy Earth Day.
Yeah.
Definitely.
Thanks,
We'll see you
Emily.
next month.
Thank you.
Thanks,
Bye,
Emily.
everyone.